So March 3rd today, topic 10, we're going to talk about respiration. So let's talk about respiration then. As I mentioned before, we were going to talk about mitochondria. I'm going to get to that in a few minutes. We're going to talk about glycolysis and a few other things first. So if you're interested in mitochondria, today is the lecture for you. And I think I promised you last day that I would have a Kahoot for the next unit. I still seem to be having some problems with it. It seems to be just working really slowly. And probably most of the questions, some of the questions may actually pertain to next time's lecture. So I'll make sure that I have it ready for next lecture. But that means we're not going to have one today. Okay, so cellular respiration. We had finished off talking about energy and ATP and enzymes. And so just want to go back and remind you why we need this ATP. And we need this ATP because our cells want to do something called cellular work. So just a quick reminder what that means. Cellular work, it means quite a number of things. So for example, it might mean pumping solutes or other products in or out of membranes. Cellular work may involve mechanical work, such as actually moving organelles or moving muscles or something like that. And then cellular work may also involve this last category. And I mentioned that this is kind of the biggest area of where ATP is used. And I'm kind of dancing around it because I'm hoping you can remember what the wording was. And I called this energy coupling. Energy coupling. And this just basically means that some reactions have a delta G that's positive, which means they need energy to proceed. And so this is where the ATP comes from. We break the bonds in ATP, and the potential energy in those bonds go towards making new bonds and new compounds. So there's our little ATP molecule. And, you know, if you're going to break it down and release energy, if you want to make it, you have to consume energy. So this is what we're going to talk about today, is how we're going to get energy and we're going to put it into those ATP molecules. And like I said, this is going to take a couple of lectures to get through this. And hopefully it won't be too painful. I'll try to go along and give you a good idea of what kind of things are expected of you in terms of what you need to know for the test. So how do we make ATP? It turns out there's two ways. And I'm going to talk about them a little bit. If you take further biochemistry courses, you get into these things in a lot of different details. And I'm going to try to point them out here today, what exactly these mean. But you're probably thinking ATP, it's made in the mitochondria. Well, yes, that's true. But not all ATP is made in the mitochondria. So technically, we have two types of ATP production. One is called substrate level phosphorylation. And the other is called oxidative phosphorylation. So notice they both have that word phosphorylation in them. So phosphorylation basically means you're putting a phosphate group onto something. So what are we putting a phosphate group on? We're putting a phosphate group on ADP. So remember, ADP is adenosine diphosphate. So we're going to add one phosphate group and we're going to make ATP. So in substrate level phosphorylation, where does that phosphate group come from? It's right in the word. It comes from a substrate. So we're going to see a little bit of this particular kind of thing happen. We're going to see this happen in glycolysis. And we're going to see it also happen in the citric acid cycle. If you don't know what the citric acid cycle is, you may have heard of it under a different name. Sometimes it's called the Krebs cycle. I'll talk about that in a minute. So you can see here's a diagram of an enzyme. You've got a substrate, it's got this little phosphate group, it gets attached to ADP, and we get ATP. What is oxidative phosphorylation? This is what is going on in the mitochondria, and this is going to involve your electron transport chain and your ATP synthesis, and we're going to talk about that. Not sure if we're quite going to get to it today. If not today, it'll be first thing next day. And this is what takes oxygen. And so that word oxidative is in there. So one way to remember what it is. So more on this later. So another thing to remind you that you may have forgotten if it's been a while since you've taken your chemistry is we're going to talk about oxidation and reduction reactions. And so what do those words mean? Oxidation means a loss of electrons. So if you take a look, I've got this little compound here, A, and it's got a couple of electrons on it, and it loses its electrons, so it's getting oxidized. Reduction is the exact opposite. It gains electrons, so we're looking at B. It picks up those electrons from A, and now it has those electrons. So my high school teacher, she taught me this little thing. I think other people have heard of it as well. Leo the lion says grr, and that's how I remember which is which. So LE is loss of electrons, O is oxidation, and grr, of course, stands for gain of electrons reduction. So just a neat little tidy way to remember that. So one of the problems when you think about oxidation reduction reactions is people learn about it in chemistry, and in chemistry often you're thinking about electrons and they float around and they're separate from the atoms. In most real circumstances that doesn't happen, and you can see in this diagram here it happens simultaneously. One compound loses electrons while the other one gains some. So it's kind of a different thing, and it's even more complex than that. But if you take a look at this simple reaction, right? This one, you know, you probably know these compounds here. We've got, or these atoms, we've got sodium, metal, and we've got chlorine. And in this case here, the sodium is going to lose an electron and it's going to become positively charged. And the chlorine gains an electron and it becomes negatively charged. So like I said, this is almost too simplistic. And we don't see this in a lot of biological reactions where things, you know, grab a charge or subtract a charge so simply. What happens a little bit more complex-wise in biological reactions is the sharing changes. So we have to think about this for a minute. Think about your basic chemistry, right? Remember, we have you've got covalent bonds and you've got ionic bonds. So let's think about those covalent bonds. Remember our covalent bonds, there were two types, right? We had the nonpolar, and that means equal sharing, and then you've got polar bonds, right? So if you take a look at our first item here, we've got methane. So carbon and hydrogen, they share their electrons in a nonpolar way, so it's kind of equal sharing. And so as methane gets oxidized by oxygen, you can see we make carbon dioxide. And so what has happened here with those electrons? Carbon still has the same number of electrons in bonds, but oxygen is a little bit more greedy. It's actually a little bit more electronegative. So it turns out that carbon is actually getting oxidized because if you look at how much sharing time it gets with those electrons, it's actually way less than before. So I know this is a little bit more you know, abstract to think about, but this is kind of the reality in terms of how the chemistry of oxidations occur. So when we say something gets oxidized, often it's involving oxygen, often it's getting its electrons are shared a little less. Usually it's being broken down into something more simple. So you might see this word a lot in biology when we talk about oxidation of fuels. And like I said, it usually means we're breaking them down, and it usually, you know, at least in some part of the reactions involves some oxygen. Not always. There are organisms that don't use oxygen at all, and so they're using something else to oxidize their compounds. So in this case, we're going to be talking about the oxidation of glucose. So you can see we're oxidizing it with oxygen. We're going to break it down into carbon dioxide and water, and this is really the thing that we care about the most, is the energy, because that's what our cells want. Our cells want energy so they can do cellular work. So not quite done with the definitions. A few more things to kind of discuss, and then we'll talk about the reactions a little bit. So even to make things just one step more, not trying to make them complicated, but like I said, you know, in biological reactions, what we're looking at a lot is sharing of electrons and swapping of bonds. And so it turns out that a lot of these oxidations in biological reactions are actually this thing here. They're dehydrogenations. So take a look at the word. D means remove. Hydrogen means hydrogen. So it's the process of removing hydrogens. So think about a hydrogen. Just ignore this. I don't know what that is all about there. Probably just a little note I forgot to delete. If you think about a hydrogen in a bond, let's say, I'm just going to draw something up in the corner there. Let's say we've got a carbon molecule up there. And we have a bond and a hydrogen. So when we say a dehydrogenation, we mean we're going to remove the hydrogen atom plus the electrons in the bond. Remember, this is 2 electrons. Every chemical bond is 2 electrons. So take a look at the chemical reaction below. We've got an organic molecule. We've got this little functional group. And we've got this other thing I'm going to talk about in a moment called an electron carrier. And we're going to remove a hydrogen. Actually, we remove 2 hydrogens. One of them ends up being free just in solution. And we end up with C double bond O. So where does that hydrogen and those 2 electrons go? They end up right here attached to this thing called NAD+. So this is a dehydrogenation. So you're going to see that word as well in some of the notes. Okay? And this is how most biological oxidations occur. It means you're scooping up a hydrogen and the electrons that go with it. So the electrons make it an oxidation because it's losing electrons. But remember, like I said, electrons don't usually actually freely exist loose out in the environment. They're almost always existing in some sort of chemical bond. So those electrons are going in a chemical bond to something new. And this new thing here is NAD+, and I'm calling this an electron carrier. It's technically a cofactor, means it's involved in the reaction. But we'll think of them as electron carriers, and that is carrying the electrons in bonds. So here's a bit more detail on NAD+. So NAD+ actually stands for nicotinamide adenine dinucleotide. So hey, we have another nucleotide. Hey, no kidding. If you look at this, we've got here's adenosine all over again, and we've got a couple of phosphates. We've got another carbohydrate, and then we have this little thing here, nicotinamide. So nicotinamide, it's derived from one of the B vitamins. Is it derived from niacin? I can't remember which B vitamin it's derived from, but it's a molecule derived from a vitamin that we have to consume as a part of our diet. So this is what NAD+ looks like. It's an abbreviation, and the plus literally means it has a positive charge. So right there's the positive charge on a nitrogen. So when we oxidize our food and we take those electrons away, we oxidize it, right? That's taking electrons. Remember, there's that word dehydrogenation. We are going to take 2 electrons and 2 hydrogens in many cases. One of the hydrogens just becomes free out by itself, and it neutralizes the charge on NAD+, and so we get NADH. So often when I think of NADH, I think of it as NAD, the acronym, and then H, literally meaning hydrogen atom. And so sometimes I actually draw it like that, just to remind myself that the H is actually a hydrogen and there's 2 electrons in a chemical bond. So I'm calling this here an electron carrier, meaning it's carrying electrons in bonds from the oxidized fuel. So in the case of respiration, we're going to talk about oxidizing glucose, of course. All right, so there's some arrows to show you where the new hydrogen ends up being attached to on NAD+. So if you take a look in your notes, there are other electron carriers out here. Here's the ones we are going to talk about in this course. Today, we're going to be talking about NAD+ and FAD, and I forgot to put the full word for FAD. It's flavin adenine dinucleotide, I believe, and then NADP+ we're going to talk about when we talk about photosynthesis. So let's get into things a bit more. So here's FAD+. So again, you've got adenosine, and we've got a diphosphate. And then we've got this flavin thing, which is from one of the vitamins that we have to consume. So it will take actually both hydrogens, and it becomes FADH2. So the H2 literally stands for the 2 H's that are attached to the new reduced form of that molecule. Okay, so hopefully you like a little bit of chemistry because we're going to get into a little bit of chemistry today. Hopefully it's not too grueling for you, and like I said, I'll try to give you an idea of what I expect you to know for this class as well. So back to this, we have two ways to generate ATP. I mentioned some of it's going to happen during glycolysis, and some of it's going to happen at that last step, which is in the mitochondria. So we'll get to that. So cellular respiration, we're going to break it down into basically 4 stages. And depending on your background, you've probably seen these stages somewhere before, maybe not necessarily broken down the same way, but we're going to be consistent with the textbook, which is mostly consistent with how most people are teaching it nowadays. I know some of your high school teachers, not being disrespectful, but some of them may be dinosaurs and they've learned it older ways and have used older names, but we'll talk about that a little bit. So first stage is glycolysis, where we are breaking glucose down into pyruvate. That happens in the cytoplasm. The second and third stages are pyruvate oxidation and the citric acid cycle. So the citric acid cycle used to be called the Krebs cycle. And depending on when your high school teacher went to school, a lot of them are still calling it that. It hasn't been called that for, I would say, at least 15, 20 years now. Most people are calling it the citric acid cycle. There's kind of a movement in the textbooks to, not necessarily to dishonor the people that did these discoveries, but to name things after the names of the chemicals and to be a bit more descriptive. So there's just been a bit of a trend like that. And so these stages happen in the mitochondria, and then the last stage is oxidative phosphorylation. That involves the electron transport chain and ATP synthesis, and that also takes place in the mitochondria. So I'm not sure if we're going to get through all these stages today. Definitely the first 3. We'll see where we get anyway. So I want to talk about glycolysis, and actually before I get to glycolysis, I'm going to, we'll do some notes here. I'm going to draw a picture for you. And I just need to share my whiteboard. Where is my whiteboard? This is my whiteboard. There it is. Okay, so I'm going to draw kind of a summary of the stages of cellular respiration. So hopefully you can all see that. There's my title, Stages of Cellular Respiration. And well, let's see here. So what I'm going to do first thing is just kind of define for you what you need to know. Okay, so 3 things to know. Okay, so when we look at these pathways, there are a lot of things going on. A ridiculous number of things. There's enzymes, there's molecules, there's water, there's carbon dioxide, there's ATP. And so the things that I want you to follow are really threefold. Okay, so I want you to follow the carbon. And what I mean by the carbon is the molecules we're oxidizing, so glucose, pyruvate, and those kind of things. I'm going to do these things in different colors. So 2 is going to be electron carriers. And 3, I'm going to do in, let's see here, I usually like to do that in red, so I'll find a red color. And 3 is ATP. So let's talk about glycolysis first. I'm going to actually draw a very kind of skeletal summary of glycolysis. All right, glycolysis, there we go. Maybe just shrink that a little bit. This is going to involve a lot of shrinking for me. You're definitely going to need a full page of paper, and depending on how big you'd like to draw, it might even end up being bigger than that. Okay, so glycolysis. We're going to take glycolysis and we're going to break down glucose. So I'm just going to draw glucose for you. So glucose looks something like this. Doing my best to be super neat for you today. It's really not easy drawing on these screens. You don't need to know the structure of glucose, by the way, not for this class, but I like drawing it. Believe it or not, there's actually something therapeutic about drawing glucose for me. Probably because I've actually drawn it, I don't know how many times in my life. So there's glucose, and glucose has 6 carbons. I'll just show you where they are right now. So we've got 1 is obvious there, 2, 3, 4, 5, and 6. So it's a 6-carbon compound. So that was one of the things you're supposed to follow, remember? So there's glucose, and glucose is going to get broken down. I'm just making a big arrow because I have a few details to fill in there. And glucose gets broken down into pyruvate, 2 pyruvates in fact. So pyruvate looks like this. So we've got 2 pyruvate molecules. And pyruvate, of course, is 3 carbons. So that's where I'm going to start. This is not everything that I'm filling out for glycolysis, but I'm going to go back to those slides and I'm going to fill in number 2 and 3, the electron carriers and ATP when we get back. But I want to go back to the slides for a minute and talk about a few things. So one thing, by the way, I just want to point out for glycolysis is we have glycolysis. So literally, glyco means carbohydrate and lysis means breaking. And like I said, we're breaking glucose from, we have a 6-carbon compound we're starting off with, and we're finishing off with a 3-carbon compound. So we're breaking it in half, in a sense. Notice, by the way, there is water involved in this reaction. I will tell you up front, I don't care about water. I just told you what I cared about. I care about carbon and electron carriers, and I care about ATP. Water is kind of one of those things. It's abundant in a cell. It's not really contributing to the energy. It's part of balance in their equations. And I'm not too concerned about balancing equations. It's useful. It has its uses, but it's not really an interesting task to balance equations. So we're not going to worry about that right now. So notice this is mentioning that glycolysis takes place in 2 phases. So we've got an investment phase and we have a payoff phase. So let me just get rid of that. There we go. And so what does that mean? It means that at the start, we have to actually invest some ATP. So what is this all about? Well, it's kind of like an activation energy. We're trying to break down glucose. It's not just going to fall apart on its own. So sometimes we have to actually put in energy before we break something down. Think of it like, let's say you're building a campfire and you need to split the wood first before you can burn it and release that energy. Same idea. We need to throw in some ATP to get the whole thing going. So we are going to consume 2 ATP. In the end, we're actually going to form 4 ATP. So if you think about the net of this whole thing, 4 minus 2 means in the end we're actually going to produce 2 ATP. We're also going to produce some NADH as well, and we'll get to that here. So let's take a look at these reactions, and we'll come back to this slide. I know this is a lot of details, but I like to kind of talk about it, repeat it, talk about it, repeat it, and hopefully by the end you've learned a little something about glycolysis. So here is the structures from the textbook. Like I said, you don't need to know the structures. You don't need to know all the enzymes, although I'm going to point out a couple here and there. But I like to look at the structures because I like to know what's going on. So there's our glucose, kind of like how I drew it for you. Maybe they drew in the hydrogens. I don't always draw in the hydrogens. Doesn't really matter. But there's glucose, and you can see in the very first step, we're going to throw in a little bit of ATP. And so ATP loses a phosphate, so it gets phosphorylated. Remember that term. And there's the phosphate there. And so we get glucose 6-phosphate. Not quite ready to break things down yet. It turns out it gets rearranged a little bit. Fructose is a little bit more symmetrical, so it's a little easier to actually split fructose than glucose. Next step is, like I said, it's almost symmetrical. You notice we have 1 phosphate, we're going to add in another phosphate, and now we have kind of a symmetrical fructose 1,6-biphosphate molecule. This is also, by the way, sometimes called 1,6-diphosphate. Bi and di both mean 2. both terms are used. So that's kind of the energy investment phase. So remember, calling back from last day's lecture, we talked about phosphofructokinase. And kind of the key thing to know is that phosphofructokinase, so phosphofructokinase, is the key regulatory enzyme of this pathway. Regulatory enzyme of glycolysis. Okay, that's kind of the key thing to know for this at this moment. So we've invested 2 ATP. So at the moment, this is not good because we've actually consumed ATP and we want to make ATP. So let's do that. So the next step is it actually basically splits in half now, and we get 2 3-carbon compounds. So we get this one here called glyceraldehyde 3-phosphate. So that is a good one to know. And the reason is because we are going to see glyceraldehyde 3-phosphate again when we talk about photosynthesis. And the other one is called dihydroxyacetone phosphate. G3P, or glyceraldehyde 3-phosphate, like I said, is important for photosynthesis because that is actually the product of photosynthesis. More on that later. I know you're thinking, wait a sec, doesn't photosynthesis make glucose? Well, yeah, sometimes. Sometimes plants don't make glucose. Sometimes they make fructose or other sugars. But they all have G3P as an intermediate. So it's broken in half. Let's get some energy out of this thing, right? We want to make some ATP. So these 2 things are interchangeable. There's an enzyme called isomerase that will interchange them. And it's actually glyceraldehyde 3-phosphate that's going to get oxidized. So here is an oxidation step, is that the glyceraldehyde 3-phosphate, here's that electron carrier. Remember, the electron carrier is going to take electrons from this thing. And I know it's not, maybe not necessarily so obvious where they are. The electrons are actually in this carbon-hydrogen bond because what they're doing is they're sneaking some phosphates in there as well. So notice everything at this point is multiplied by 2 because we had 1 glucose, and that glucose was broken down into 2 3-carbon molecules, and so everything is multiplied by 2. Don't worry too much about the stoichiometry, but that's why there's 2 in front. You can see 2 here and 2 here and so on, right? I'm just explaining what's going on here. So now we have this funny little compound here, this biphosphate glycerate, and we've made a little bit of NADH, which is good. We're going to break this down a bit more, and now we're actually going to make some ATP. So this is great. So here is our first ATP being produced, and the way it's being produced is that term that we called substrate level phosphorylation. Yeah, so we've made 2 ATP at this point. So at this point now we're at a 0 gain sum, right? Meaning we consume 2 ATP, we've made 2 ATP, but we're not done. We're not done glycolysis. So a little bit more, there's our 3-phosphoglycerate, and it gets rearranged a little bit. You can see there's some water going away. None of these steps here really are too important for this class here. But that final step, so notice we have this penultimate compound is PEP. We mentioned that last day as a regulatory molecule for the phosphofructokinase, and it has a phosphate on it, and so we are getting some more substrate level phosphorylation. Substrate level phosphorylation. So we've made 2 more ATP, and this is great, and we've made it all the way to the end. We've made it to pyruvate. So that's 10 chemical reactions, if you can believe it or not. 10. If you go on and you do biochemistry, you're going to have to learn these reactions inside out and backwards. It's not as bad as it looks. There is some logic to what's going on in terms of the chemistry in these things. Okay, so there is the payoff phase kind of all in one picture, and you can see we've made a whole bunch of good stuff here. We've made some ATP, we've made some NADH, and we've made some pyruvate. So I'm going to go back to the whiteboard, and I'm going to fill in some of those details. So here's our glycolysis. So we'll talk about those electron carriers first. So the electron carriers, we had 2 of them. We made 2 NADH. So in order to make NADH, we need NAD+, that's the ingredient, and we make 2 NADH. There we go. So I know you're thinking, so what's so important about NADH? Remember, these are eventually going to go to the mitochondria, and they're going to be involved in that electron transport chain. And so ultimately, those are going to be used to make more ATP. And ATP is what we care about the most. So for ATP, I'm not going to talk about putting in 2 and you get out 4. I'm just going to talk about the net. And so the net is we're making 2 ATP. So to make 2 ATP, we need 2 ADP. And we need 2 of those little inorganic phosphate things, so we call that PI. And then we're going to make 2 ATP. So there's kind of glycolysis in a nutshell. If you're going to learn any one thing about glycolysis, this diagram is the place to be, okay? Everything else is details, but you know, focus on this. You've got a reactant, you've got a product. Actually, you've got several reactants and several products. Water's involved, but we don't care about that so much. And it's important that you know kind of these basic details around every reaction. When we look at between respiration and photosynthesis, I'm just trying to count in my head here, about 5, 6, 7, maybe 7 reactions we're going to be looking at overall. And here's something about midterm 2, is on midterm 2, I'm going to give you a pathway. And so it could be a picture of glycolysis, or it could be a picture of the citric acid cycle, or the Calvin cycle in photosynthesis. And I'm going to ask you some questions about it. And the first question is going to be, what biochemical pathway is this? And so if you know this, if you know that, okay, wait a second, it starts off with glucose and it finishes with pyruvate, then right away you can say that is glycolysis, and you can get the first mark for that question. Okay, so I'll try to remind you again, but it's important to know this amount of information about these pathways. So we'll come back to this diagram. I'm just going to go back to the PowerPoint and finish talking about glycolysis. I told you I'd come back to this, and hopefully it makes a little bit more sense in terms of what is going on. We've got the investment phase where we are consuming 2 ATP. We've got the payoff phase where we are producing 4 ATP and we're producing 2 NADH, and we end up with pyruvate. And so you can see the net reaction at the bottom. They're being a bit more thorough than I am because they have the water, and they're doing 4 minus 2 for the ATP. So that is glycolysis. And thought I had, I'm looking for a different picture. Glycolysis takes place in the cytoplasm. And so maybe that's worth it to note. I'm going to go back to this diagram here. And this is where I'm going to probably try to shrink and move things around a little bit. So here we go. I'm going to just draw right here a little dotted line. And there we go. And so now at this point, we are exiting the cytoplasm. And we are going to enter the mitochondria. So you can just pretend that's the mitochondrial membranes. Mitochondria. There we go. And that's where the next steps are going to take place. So back to the PowerPoint here. I know I have some slides on mitochondria, but maybe it's after pyruvate oxidation. So we'll see here. So the next step is pyruvate oxidation. And this is one chemical reaction, but it's kind of like crazy how many things are going on. And I apologize for that, but I didn't invent it. There's actually a massive enzyme that does this, and it does a whole bunch of things at once. So let's kind of pick this apart. First thing to know is that this happens basically in the mitochondria. You can see the enzyme itself is I believe it's actually associated with the inner mitochondrial membrane. So basically pyruvate is transported into the mitochondria, it loses the carbon dioxide, it picks up coenzyme A, and it produces an NADH. So let's talk about this a bit. Maybe it's worth it to talk about coenzyme A here. So what is coenzyme A? So coenzyme A, again, you could ignore this here, but basically saying it's from a vitamin. We've got adenosine all over again. Look at that. There's adenosine. And then we've got a bunch of other functional groups on there. And so this is a coenzyme, meaning it works with an enzyme, it's modified, but then gets modified back right away. So coenzyme A is sometimes abbreviated CoA, like that. And sometimes, even more frequently, it's abbreviated as CoAsh. So the ash actually stands for SH, meaning sulfur and hydrogen. So that's what it looks like. And okay, there's some labels there. I didn't realize I had the labels. We're going to make something called acetyl-CoA. So there's the sulfur over here again. And this here is actually an acetate group. And that's what we're going to remove from pyruvate. So what's the big deal here? Like, why are we, this seems really complicated. We have this giant molecule grabbing onto a couple of carbons. Well, you can kind of think of this as a bit of a courier. And what it does is it basically transports that carbon from one stage to the next stage. Because this carbon here, it's not easy to break down. And when you have two molecules, two atoms, it's a little bit easier to, if you have a hold of it, right? Like some of the small things are kind of hard to get a hold of, and that's kind of how I think of it. It's just grabbing onto it for a minute is really what acetyl-CoA is doing. So there's the acetyl group. So I want to draw this reaction a little bit on the whiteboard, and like I said, we'll talk about what's going on a little bit as we draw it. Okay, so the second reaction here, I'm going to do my arrow. It's going to be kind of a little bit on the long end so I can fit everything in again. And this stage is called pyruvate oxidation. I have seen at least one textbook call it pyruvate, what do they call it? Pyruvate decarboxylation, which means it's losing a carbon dioxide as well. So what is going on here with the carbon? So the carbon, we're adding this coenzyme A. So there's my coenzyme A. And remember, we're calling it CoAsh because it has the sulfur and the hydrogen. So that is going in, and just make it tiny bit smaller so we can squeeze it in here. And we're going to make acetyl-CoA. I can draw acetyl-CoA for you. It's not really too hard. It kind of looks like this. So we've got the S and the coenzyme A, and then we've got, if you think about what's going on with the pyruvate, we still have the bottom part of pyruvate. So take a look at that. What are we missing from pyruvate? We are missing a carbon and two oxygens, and that, of course, is going to get released as CO2, carbon and two oxygens, so carbon dioxide there. Okay, there's no ATP produced at this stage, none whatsoever. But what we are doing is, oh, by the way, just to keep some tally on this whole thing, I'm gonna actually, and I am personally gonna keep track of the numbers here. So we're actually making 2 acetyl-CoA. Everything here we can multiply by 2, by the way. The last thing, of course, is the NADH. We are oxidizing a little bit. So NAD plus comes NADH. Like I said, I can put a 2 in front of all these things. I'm just doing it for my own sake so I can remember what is going on here. And I guess I meant to draw that in blue. So let me, I'll rewrite that in blue. Hold on. I like to color code things. All right, 2 NAD plus and 2 NADH. There we go. So that's pyruvate oxidation. Like I said, it's basically getting pyruvate ready for the citric acid cycle. It's Looks complicated, and in some ways it is. There's a few things going on, but it's just a further oxidation step and releasing a little bit of carbon dioxide. Okay, so it says here this is the junction between glycolysis and the citric acid cycle. So we do want to talk about the citric acid cycle as well. So there is, here's where we're at. We're in the mitochondria now. And so I want to talk about mitochondria for a few minutes, and then we'll talk about the citric acid cycle. So here's the mitochondria, the mighty mitochondria. We have talked about them a little bit already, and I mentioned, you know, they have their own DNA, they have their own ribosomes. And what I want to do is usually at this stage is, oh boy, I'm missing, my notes are not organized the way I thought they were going to be. Hold on a second. Yeah, okay. I guess I thought I was going to draw one for you, but that's a little bit later on, and I'm not sure if I'm going to get to that today. We'll see anyway. So the mitochondria, like I said, I'm gonna draw a picture for you. Maybe it'll be a good way to do some review next day. But take a look. We've got an inner membrane, and we've got an outer membrane. So the outer membrane there is basically structural. It's just holding the whole thing together. And the inner membrane has all those folds in there. And the inner membrane is super important for the electron transport chain, which we're going to get to next day. And the innermost part is called the matrix. And it's kind of funny, sometimes in class I ask how many people have seen The Matrix. And, you know, it was a pretty cool movie in its time. And of course, you guys are all so young that you were probably born after The Matrix even came out. But it was on Netflix recently. So if you're interested in a cool kind of kung fu techno movie, it's a pretty cool movie. So Matrix, this is where we're going now with the citric acid cycle. So here's the citric acid cycle. You can see up at the top we have pyruvate oxidation. We're going to take acetyl-CoA, and we're going to break that down, and we're going to make some more NADH, we're going to make some ATP, and we're going to make that other electron carrier, which is FADH2. All the carbon is going to get released as carbon dioxide. So let's take a look at what's going on here just in a bit more. Okay, that's like the same slide, so I apologize for that. Let's take a look at what's going on in terms of the chemistry, right? So there's our acetyl-CoA. It's 2 carbons. It releases the coenzyme A right here. And we take a 4-carbon compound called acetyl or oxaloacetate, and we're combining it with the acetyl group, and we're getting a 6-carbon compound. So I don't know if anybody here likes sour candy, but this is what makes your sour candy sour, is something called citric acid or citrate. So thank you for sour candy. This gets transformed a little bit isocitrate. You can see a little bit of isomerization going on. Not really a big deal. But the next few steps are a big deal, because you can see here we're making some NADH, so we're oxidizing it. It's losing some electrons. It's also losing some carbon. And then we're going to do that again, lose some more carbon, lose some more electrons, and produce more NADH. And then we're going to make some ATP. So I know you were thinking, oh, we're going to make some more NADH. Not yet. So it turns out that we actually make GTP instead of ATP. So you're probably thinking, what is going on there? Well, okay, here's some news for you. ATP is not the only energy source we can use in our cells. We do have other nucleotides, right? We have the all the codes. We've got the genetic alphabet, right? We've got the G and the C and the A and the T and the U. So ATP just happens to be the most commonly used nucleotide energy source. GTP is the second. So some enzymes use GTP. You can see what they're showing here is that the GTP can be easily converted to ATP. So sometimes in textbooks it says ATP is made, sometimes it says GTP. In this case, they're trying to show you that both is possible. We've got another step. We've got another oxidation step. We're making some more electron carriers, and we're making some more. So 10 more reactions, right? Or sorry, 8 more reactions. So a lot going on here. Mostly it's oxidizing the thing. It's stripping the electrons off. It's releasing the carbon. And as a bonus, we're making a little bit of ATP. So let me summarize this for you on my diagram. Okay, I know this is getting smaller and smaller. I'm trying to fit everything in here. All right, so I'm going to slide this over. Sorry if it's, okay, come on, just slide over for me. There we go. Okay, so now we're going to do the citric acid cycle. And so what I'm going to do, the citric acid cycle, is just going to look like this. Okay, so citric acid cycle. There we go. So what did we say was going on? Well, the acetyl-CoA gets released, so we're going to release the acetyl-CoA. So remember that's also known as CoAsh. Okay, I'm not going to draw all the other carbon compounds. There are a whole bunch in there. Citric acid being maybe the most notable because it gives us sour candy. There are a few others in there, but they're not really that important in some ways. What else is going on? Well, we are releasing carbon dioxide, so I'm going to just put that over here, release it over there. I'm going to just make this a little bit neater here. Okay, there we go. Okay, so 2 carbon dioxide are getting released. Actually multiply that by 2 again because we have 2 acetyl-CoAs going into this. But we'll tally things up here in the end. We're going to make some NADH, and we're going to do that 3 times. So we're going to take 3 NAD+. Okay, sorry again, I've meant to do that in blue. Here we go, here comes the blue. 3 NADH. For NAD+, make 3 NADH. And what else were we doing? We made some ATP, so we're going to do that here in red. So to make ATP, we need ADP plus PI. If you want to change this to GTP, I am totally fine with that, because that is actually more accurate. But ATP is what we care about the most. And then the last thing is we're making one more electron carrier, and remember it's a different electron carrier this time. It's called FAD, and it makes FADH2. So there's all the things we care about: the carbon, the ATP, the electron carriers, and we've got the citric acid cycle. So I'm just going to wrap up here in a minute. I have a couple other things to say. I will probably come back to this diagram. Maybe not necessarily the whole thing, because this is pretty much mostly what I want to draw for the stages of cellular respiration. I know there's one more stage left. We still want to talk about oxidative phosphorylation and the electron transport chain, and that's kind of not so easy to draw. And that mostly just involves all the blue things, all the NADHs and the FADH2s. So if you want to make this complete, you could take every NADH and put to the electron transport chain, to the electron transport chain, to the electron transport chain, and then one more right here, to the electron transport chain. And then basically that diagram is complete. So hopefully that turned out nice for you. I just have a couple more things to say about the citric acid cycle, and then we'll wrap up for today. So there's the citric acid cycle in all the gory details. And like I said, kind of really the important thing to know is our end products. And here we go. We've made some NADH, we've made some FADH2. We're releasing some carbon dioxide. That's just a waste product at this point, so not really that important. And making a couple of ATP, and we like ATP. In the end, oxaloacetate is reformed and ready for another round of the cycle. So all you need is a little bit more acetyl-CoA, and then away we go. Here's kind of the simplistic version of the whole thing, the bare bones version, which I just drew for you. And like I said, this is kind of the main thing to know. So think about what we've tallied up here. We have glycolysis. We've made 2 ATP up there. Pyruvate oxidation, we've made no ATP. Citric acid cycle, we've made 2 more ATP. So it says not much energy yet, or is there? That's because all of these NADHs, you can see these arrows here, all of these things are eventually going to go to that last step where we are going to make a whole bunch of ATP. And that is what we're going to talk about next day, is the electron transport chain and chemiosmosis and how we're going to make all of that ATP. So thanks for coming out. Hope you have a wonderful Wednesday, and I will see you Friday afternoon.